Hi, everyone. Welcome to this very special session, the announcement ceremony of the first edition of the Longevity Prize, which is the Hypothesis Prize. I'm so delighted that many of you joined. I'm even more delighted that we are finally in the final stages where we can actually announce the winner. I feel like it's been quite the roller coaster since the first time when VitaDAO had the great success of getting their hands on this really wonderful quadratic funded grant, and that had reached out to a few collaborators, including Methuselah Foundation, including Lifespan.io and Foresight, and to really get this project rolling. So thanks so much. It's been such a fun collaboration. Thank you so much for all the judges in the room that judged the prize. And obviously, thanks to everyone who submitted their proposals. The first edition was, I think, just really magical. I'm really delighted to have been a part of it and to have read so many really wonderful submissions. And I really do mean this. It was a true delight to look at all the really wonderful research out there. So thanks everyone for putting so much work into this. We're all kind of donating our time here, and I think it really paid off. Okay, well, that's all from me. I'll be in the chat later on. I'll be moderating a little bit through the Q&A. I'm Alison from Foresight Institute. And I'm really delighted to hand it over to Vincent to maybe describe a little bit more what this prize is actually about. I'm also going to share some documentation here in the chat if you're live with us on the call, and otherwise it will be in the YouTube description. So thanks everyone for joining. I'm really happy that we get to do this and super excited to announce the winners.
Yes, so super excited for this prize. I want just to give some background on how it came about. So basically, also together with Keith from Lifespan, who's also here, and myself, and I'm with VitaDAO, we basically organized, I think almost 2 years ago, a donation round for longevity projects called the Quadratic Coin Donation Round, with the anchor donor being Vitalik Buterin, who came up also with this idea of quadratically funding public goods and nonprofit projects. And basically in this round that we organized 2 years ago, I think about 60 projects participated, different open science and open longevity efforts. And one of them was also that we participated with was the Longevity Prize, which was just like an idea that I had with others at VitaDAO, and which ended up getting over 8,000 donors in this donation round. And basically because of our quadratic donation work, it ended up with a big donation of over $200,000. And also, of course, a lot of micro donations from these 8,000 donors that I also want to thank for making it happen. And afterwards, we basically Keith joined, and Alison with Foresight, and Dane with Methuselah, and many others also from the VitaDAO community, from our extended community as part of the jury, and started proposing prizes. So the idea was to not make it one gigantic prize like an X prize, which is also amazing, but we wanted to supplement it with more small prizes, like this first one, which is an idea prize that was proposed by Sebastian Brunemeyer. So actually a lot of the jury member and everyone can propose a prize. And we actually had over 40 prizes proposed, a lot of them smaller prizes, already a second one in the pipeline with a biomarker prize and a generic drug repurposing prize. And there will be more prizes. But we wanted to start off with this idea prize to really generate as many interesting ideas on how we could solve longevity with kind of obscure and underexplored areas. So big thank you to Sebastian. I'm not sure if he's already in the call, but otherwise I would. Okay, so I can give the high-level intro on the Hypothesis Prize itself. So basically, Sebastian came up with this idea for the Hypothesis Prize. And the idea was to highlight and generate ideas for the most promising but underappreciated obscure areas of longevity science that we should pursue. And he made an example of heterochronic parabiosis, it being one of them, that was, of course, revived by Irina Boy in the 2000s and is now one of the hottest and most difficult areas to perform. But that was not really known, or that was a bit neglected maybe. And he wanted to surface more of these ideas. And we were delighted to see more than 100 applications of different ideas coming in on this prize, and having 3 winners and some honorable mentions to announce for this call. So that's it from me. So to hand over to Dane or to the Dome, probably, right? Like if our special guest and community.
Are you there?
So basically, for context, The Dome is an event location at kind of pop-up city that was organized by Vitalik, who is also the anchor donor of this prize, even though indirectly to enabling this Gitcoin donation round. And they would join us for a brief message, but I think they will connect their whole auditorium to this conversation, so they might still be in the process of doing so. So we should give them another minute probably. Okay, they'll take another 10 minutes, so maybe we'll hand over to Dane and then they can join after.
Okay, cool. So I've only got 10 minutes to fill. So good thing I have access to the English dictionary. Aardvark, ameliorate. Anyway, so I can't do that. But anyway, it's great to see you all. Looking forward to seeing everybody at Zuzulu. I wore swimming trunks today in your honor. Looking forward to using those as much as possible in a couple of weeks. As far as this challenge goes, I'm extremely excited to be part of it. When we heard about it, basically the second that Allison said, "Hey, we're doing this thing," I was like, "Oh, great! I'd love to do it." So we got started about 20 years ago because we wanted to do two things. We saw aging as a disease, and we thought it was crazy that more people weren't thinking about it that way. And two, we liked prizes, and so the longevity prize was kind of a good thing for us. Prizes are really, really fantastic when the market doesn't get innovation to happen the way that it should. And so if you put a prize out, it tends to get people to do the work that they need to do. And we've been doing prizes now, a number of different prizes for several years. We got started with the Methuselah Mouse Prize long time ago, right around the same time that X Prize was kicking off. And then in the last few years, we've been working with NASA on a couple of different challenges. One around tissue engineering called the Vascular Tissue Challenge, and then another one right now on nutrition called the Deep Space Food Challenge. And I love the longevity prize, especially its kind of semi-decentralized nature. A lot of times challenges, when they're put together, they aren't designed in as collaborative a manner as they could be, and they're not judged in as collaborative a manner as they could be. And so it's really, really nice that this is, to me anyway, this, and I think everybody probably agree with this, that this challenge is really, the longevity prize is really, it's a community effort. Most of the time when challenges are put out, they're either put out by like monolithic entities, or they're put together in such a way where there's almost no control for the community to kind of like speak their mind and represent themselves. And I think it's really, really cool that this one is much more inclusive in that regard. And I think that the sort of stuff that we're going to be trying to do in the future it's around that, both in terms of idea generation and in prize design itself, but also in the sorts of activities that can run alongside it. So Vincent briefly mentioned that the biomarker challenge that we're working on. So that was kind of a funny one. That was put out by Aaron King in the initial prize proposals as a database challenge. And we were like, oh, that's really, really perfect. We had been working on, and it was going to be much more focused on clinical aging biomarkers, a musculoskeletal system, that sort of thing, less aging clocks. And we thought that was really, really great because we couldn't find any kind of centralized repo for that sort of thing. And so at Methuselah, we had been doing our own kind of research on that for a while. And then a few years later, we saw that pop up and we're like, hey, that's maybe a thread that we could start pulling on again. And so we agreed to put up 50K for that. But as soon as we started developing it, I had a friend of mine who was working on the development of that challenge, and he met a guy in Dublin named Madi Mokri, who happened to be kicking off this biomarker consortium. And I was like, well, we don't need to do this now in the way that we had originally thought about it. So we got in touch with him, and as the first manuscript was being written, it was very helpful for us to change the way that we were thinking about designing it to be something that was more, it was kind of a split thing where it's both about validating aging clocks and those sorts of standards, which are, frankly, they're useful they're certainly useful as heuristics, but the consumer facing ones are sometimes not so great. It's kind of a mixed bag. And so it's really important now, given the state of the industry, the community, there's a ton of products that are likely to be coming to market in the next 5 years or so. So it's really important for us to have a process that we're engaged in that lets us do consensus reasonably well. And so the consortium is really, really important, I think, for that. And the outputs from that consortium are going to inform very, very fundamentally the core of what the biomarker challenge will be. So thankfully, Madi put together an incredible crew over there. We've got Andrea Meyer and Eric Verdin and Michael Snyder and Vittorio Sebastiano and Vadim Gladyshev and a gazillion other people. I mean, it really is kind of an embarrassment of riches. So I don't know if Madi is even on this call, but thanks, Madi, for putting that together. I think it's going to make it much easier for us to develop a biomarker challenge that's, truly representative where the science sits, and also is very inclusive in terms of pulling in so many people, like real proper subject matter experts from the community, people who've been around this, have been in this field now for a very, 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 very long time. So I'm happy that that's happening. And then I don't know how many more minutes do I have to talk before I start about my childhood? Just 1 minute? Okay. So the other thing that we're doing that I'm excited about is we're developing a I guess you could call it, we're calling it animal-free precision medicine, and it's in response to the FDA Modernization Act. The hope being to dramatically decrease the amount of time and the cost it takes to do clinical trials, because it really is absolutely insane that we have to rely on animal models so often, and that the clinical trial costs 50 million bucks to do. That's absolutely freaking insane. So that's a bigger challenge. That's going to be at least $1 million, and we've got a number of partners that we're in talks with, but it's still somewhat early, but we're getting close to having the broad stroke criteria put together. Well, that's actually ready, but we're cleaning it up a little bit before we share with the community. But we're going to be probably reaching out to a whole bunch of people on this call to get their feedback so that we can develop something that's truly thoughtful and with respect to what we're looking for as a community.
I think we could maybe give Saba like 1 minute to share kind of about like one of the third prizes in the working of the drug repurposing prize. I can ask you to unmute Saba if you have audio and want to speak about it for a minute.
Yeah, sure. Thanks, guys. And thanks, Dane. Yes, this is actually kind of tied into the biomarker prize as well. But the idea, what we're really interested in, is there's a lot of off-patent drugs, like generic drugs, that could be very effective for improving healthy longevity, but they lack a private incentive to fund clinical trials, because basically everyone can buy these drugs for very cheap. But that's also a positive as well. It means everyone can benefit from these drugs. So things like rapamycin, metformin, NMN, even things like dietary restrictions and interventions. But there's a lack of incentive to do clinical trials unless you can enforce a monopoly price. And one of the things that we've been involved with our organization, Crowdfunded Cures, is about basically using prizes as an alternative to patents. And obviously that's something Methuselah Foundation is also very heavily involved with, and we've sort of been chatting about that for many years, actually. But basically like a mouse prize, but in humans and for clinical trials. So that's something we're really interested in, and specifically for generic drug repurposing. But happy to chat with any folks that are interested in that.
Awesome. And what I just wanted to add, what I also threw in the comments, maybe for the end of this call, we have this Google document with different proposed prizes. So already like the up to 22 prizes. We actually had 50 and already sorted out some that weren't worked out. But if you want to add ideas or even comments to the proposed prizes, feel free afterwards to jump in the Google doc.
Yeah, that's a great idea, because we found that the community is very good at spotting problems. And so whether we turn something into a proper prize or not, or it's something that's a really fantastic product idea, right? Like it's a really, really great repo that we can then use to spin up other activities and react in terms of the way that we structure these sorts of challenges. So yeah, that's just echoing you, Vincent.
I think the Dome is joining us, but we can't hear you yet. So you have to unmute maybe, Lawrence.
Yeah. And if you can turn your camera on too, that'd be pretty neat.
Can you hear me?
I can hear you.
Yes.
Yes.
All right. So we have a camera on this account, this other account.
There's two screens, Zuzulu Dome and VitaDAO Longevity. You're on the second panel to the right. So you might need to bring it to the main screen or get rid of Zuzulu Dome sessions.
The Zuzulu Dome will be the audio and the other one will be the video.
See us now? Oh yeah. We see a VitaDAO Longevity. There you go. Hey, Lawrence.
All right, so I'm gonna put the camera here. Vitalik, take it away.
Okay, great. Yeah, thank you. So
I guess it's here to announce an entry and talk about prizes. And prizes for public goods funding are a thing that I've been following closely for a long time. Like being each. I remember being introduced to the concept by people like Alex Tabarrok and Tyler Cohen, the economists over at GMU, when they're talking about prices as a possible alternative to things like patents for funding innovation. And within the Ethereum ecosystem, we've been experimenting with different kinds of funding models a lot. And we've been doing retroactive public goods funding quite a lot recently within the optimism ecosystem. Also, things like the Protocol Guild, which is certainly a prize in some sense. So, like basically the idea of all these mechanisms, right, is that instead of funding a specific team proactively, with the funder also having to decide which team is the most likely to accomplish a particular objective, you're funding an objective, right? And you're basically saying, I'm willing to give money to whichever team does the best possible job of achieving that objective. And that approach has some really nice benefits, basically because it works well in the context of funders that have lower information, because the funder does not have to choose the team. They just have to choose the objective. And to the extent that the objective is a subjective one, they have to two judges, but that's still much less subjective than choosing the best team for the job. If the funder does not fully trust itself to be incredibly neutral, if the funder does not trust itself not to be corrupt, for example, as an organization, then prizes again have the benefit that they reduce the number of dimensions available for people to give money to themselves, which is really nice. So they have a lot of benefits, and I think they have a lot of benefits that are particularly interesting in the context of this intersection with the crypto space and DAOs and incredibly neutral protocols and all of these things. But prizes also have some weaknesses, right? So like one weakness, for example, is that specific teams do not have certainty that they are going to get money, because specific teams don't have certainty that they're going to win. Teams don't get the reward until the end, and maybe teams do need funding at the beginning, right? Sometimes the objective really is so subjective that the problem of measuring whether or not it has been achieved is as hard as the objective of deciding whether or not the work that a particular team is doing is good, right? Like if the Ethereum ecosystem, for example, wanted to create a prize for the best community organization in Latin America, just to give one example. Like, what would best be? Like, what are the goals that are kind of hidden in that? Like, the process of retroactively judging kind of what the best community organization is, well, that's going to include as many kind of so many different knobs for people to kind of maneuver that it doesn't actually it's actually not that far from choosing it. Right. But on the other hand, if you try to be more formal and choose specific metrics, then there's good hard law, right? Like every metric ceases to be a good metric once you try to use that metric as a target, right? Even if a particular measurement corresponds to what you actually want in the normal world, once you put a huge amount of pressure to optimize for that specific thing, well, it often ends up overused by it, right? So some of these things are things that need to be improved inside the mechanism, right? So in the case of Optimism RPGF, for example, Optimism RPGF, they've been experimenting with kind of judging mechanisms for deciding which projects contributed the most value, trying to create more and more norms around that. Another thing that you can do is you can kind of combine the prize-making institution or the retroactive prize-making institution with proactive investment, right? So if I invest in a team and that team ends up winning a prize, then I, as an investor, could get a share of that prize. And this could create an ecosystem where investors basically get incentivized to be predictors of which team does a good job, and they can specialize on that. And the group that's giving the prize just needs to focus on the much smaller and more contained task of figuring out whether or not they should give the prize and which team actually got it at what time, right? So there's a lot of interesting challenges, and the crypto space, I think, has been a great kind of beginning testing ground for a lot of these issues, and I expect it will continue to be. And I think the SI and the buyer space is also a really important one, right? It's an industry where there's a lot of different problems. It's a space where there's a lot of desire, I think, to actually try to be open and include different teams and try to make mechanisms that are open to teams that come from a kind of more non-traditional contexts. There's a desire to try to even kind of start from objectives and identify objectives that could be stepping stones towards something more helpful. There's a desire to find structures that provide funding to projects who need it, but that at the same time preserve the incentives for things like openness and collaboration. And so I think it's also an environment where prizes are really good to experiment with. So, right, that can come in many forms, right? So you could have topic-specific prizes. So the hypothesis prize is something that we're going to get some winners announced for. Then just a retroactive public goods funding rounds for longevity-related stuff in general, right? Like if you make a biomarker and that biomarker turns out to be the best one 5 years from now, then that could be something that gets retro funded. Also, not everything has to be retro. You can also have proactive funding rounds in longevity as well. So a lot of different opportunities, and I'm excited to see VitaDAO and the DeSci and biology ecosystem in general experimenting with these things. And so I'm excited to see what people come up with.
All right. Well, that was amazing. So you preaching to the choir, man. That was very good, very thorough. That was a fantastic crash course on prize economics and all the things that are wrong with traditional prizes, because there is no shortage. But there's a lot of really, really great stuff that we can do. I'm really interested in particular in you mentioned the retroactives, public goods. This is something that we're definitely looking into a lot with regard to finding ways to distribute capital more thoughtfully across the life cycle of a challenge. Because yeah, you can put it out there, but most of the time, the cost to even enter into the challenge, the cost to do the work necessitates them going out and finding money from someplace else. And it makes it very difficult for them to focus on the exact thing that you were looking for in the first place. So you end up with lots of time, really, really great work being left on the table as a result of that. So anyway, now to the especially fun part. The real star of the show today are the winners of this thing. They worked hard to put together these proposals. And I want to mention too that even though we asked for short proposals to make this easy for folks, there's an enormous amount of the work that goes into architecting what those are in the first place. So in a way, like this is the Cliff Notes version, right? And then there's tons of documentation and publications that they put together that either they worked on themselves or they cited or whatever it is that we then went through in the screening and in the judging panels to go through and evaluate what seemed to be of most value. So I'm going to go ahead and get started. And we're going to do it backwards today, because even though there's first, second, third place and honorable mentions, just because we had to pick some numbers, this really is a community effort. So we're going to start with number one and work our way around. So for first place, I gotta stop, is Carlos Galicia from C and the Buck Institute. And he worked on, the title of the submission was Harnessing the Rejuvenation Process that Occurs with Embryogenesis. And it was a very, very cool throwback to some work from Tracy Sonborn in 1930. So it's very nice to see. So if we can turn the mic over to him, is Carlos around?
Yes, hi, I'm here.
All right, take it away.
All right, so yeah, I have a short presentation of my proposal that I would like to share with all of you. Let me share this. But first of all, I would like to thank all the people that are involved in this, right, and have given us this opportunity to share our obscure and hopefully promising areas of research directions in the longevity field. And getting into my presentation, so the title of my proposal was Harnessing the Rejuvenation Processes that Occur During Embryogenesis. And when I think about what are the most promising areas in longevity research, I like to think about how evolution has dealt with aging, right? And this is a very species-specific answer. So, for example, this is a picture of the immortal hydra, right? So probably the ultimate example of dealing with aging. And the way the hydra does it is it constantly creates new cells that go on, differentiate, perform their functions, and then the animal discards these cells, and new cells are generated again. And this is a constant process. And this is the mechanism that is thought to give hydra its immortality. And in humans, we don't really do that. We don't have a mechanism like that. But what we do is we have the process of embryogenesis. And embryogenesis starts with two cells from adult individuals, the gametes, and they come together and they form an embryo that undergoes a rejuvenation process that gives rise to a new young individual, right? And the central idea in my proposal is how can we harness these rejuvenation processes that go on during embryogenesis? And to do this, I propose 3 different phases. And the very first phase of the proposal is to characterize and understand how gamete aging works. And I think this is really important because before we can study how the embryo is actually performing rejuvenation, we need to understand what is it that the embryo is prepared to rejuvenate. And a way to understand that is to understand what are the age-related changes they accumulate in the gametes and the embryo has to deal with. And the way I propose to do this practically is actually pretty straightforward. So it's just taking gametes from organisms at different ages and do very deep phenotyping and characterization of the cellular components of these gametes. And these are some of the examples of the things that I mentioned in the proposal. So, for example, characterizing measurement of the soluble and insoluble proteins, RNA sequencing, metabolite and lipid profile, organelle function, as well as covalent DNA modifications. And in covalent DNA modifications, it's included things like methylation, right, which has given rise to the methylation clocks. But in reality, there is a ton of different covalent DNA modifications that occur in organisms and that are passed down to embryos, right? And they have to deal with them. So once we do this, we can have a good idea of what's changing with age in the embryos and what types of damage and other processes are changing. And then the next step is to follow these hallmarks of gamete aging through embryogenesis. And this will allow us to learn how the embryo is actually dealing with all these age-related changes that occur in the gamete. And it's, again, a very simple and similar project for this. So it's just taking embryos at different stages of development and, again, doing this deep characterization and phenotyping of the cellular components of the embryo. And what this will allow us to do, it will allow us to identify at what point in embryonic development different rejuvenation mechanisms are getting activated and are dealing with the age-related changes that occur in the gametes. And it will let us know what genes and proteins are being expressed at that specific point, right? So once we have this model of aging and rejuvenation, we can start to try to find therapeutic opportunities. And the way I see this occurring is we have this temporal model of the things that happen with aging and the things that happen during rejuvenation. And how we go specifically about this is very dependent on what we find in phase 1 and phase 2. But I can give you a couple examples of the way I'm thinking about this. So, for example, if we were to identify a transcriptional program that is highly correlated with a specific rejuvenation event, we can try to identify what are the genes that are actually causing this transcriptional program. There are a number of computational methods to try to identify small molecules or ligands that produce a transcriptional response similar to the transcriptional program that you're interested in. So there are different ways in which we could try to find therapeutics. But I also think that it's really important to understand what are the conditions necessary for rejuvenation. So, for example, something that I think about a lot, and I think we don't have a good grasp of it, is how necessary is cell division for rejuvenation to occur? You can imagine if you have a cell and it has accumulated different types of age-related damage, we don't really know if we have all the mechanisms necessary to deal with all the types of damage that occur during aging, or if sometimes just the mechanical process of dividing the cell is necessary to dilute the damage, right? And having this temporal model give us information about that. You can imagine if you have a cell, an embryo, when it's one cell and it has this X amount of damage, and then when it has 2 cells, it has half of that, and when it has 4 cells, it has a fourth of that. We can infer that these types of damage are probably requiring cell division to occur to be diluted to the point where they are not relevant anymore, right? And this is valuable information that we are going to need if we want to keep moving forward these therapies that are based on mimicking what the embryo does, like, for example, genetic reprogramming. So yeah, that's my presentation. Thank you all again. And this is my Twitter and email. If you have comments, questions, or would like to chat about the details, feel free to reach out. Thanks.
Cool. Thanks, Carlos. Yeah, that was awesome. I really appreciate it. Yeah, and I want to mention too that in terms of the way that the winners were chosen, we looked at both the distribution and the average score and normalized score internal to each individual judge, but also we looked for something like a semi-consensus between different types of folks within the judging panel. So we had academic SMEs, and then we had people that were more on the industry side or that were investor sorts of folks. And the winners and the honorable mentions had a really kind of very telling distribution in terms of they were both very, very heavily liked by the academic side of the community and also on the commercialization side of it. So I just wanted to call that out. And thank you, Carlos, again for that awesome presentation. So we're going to go now into the number 2. So our second place winner is Rakan Aimbetov from VitaDAO, and he did some work on RiboAge. So hopefully he's around too, so that he can talk for 5 minutes or so.
Yes, sure. Hi,
Yes,
everyone. Yeah, so let me put up my presentation. So basically, my proposal is devoted to protein glycation. So basically, the glycation of ribosomes. And
We
how
see a wave, a blue-orange wave. Is that correct?
Not really, because I'm sharing my presentation and it should be visible. You don't see anything, right?
No, if you close the window, then
Oh, well, wait.
the presentation, and if it's the last window that was opened before Zoom, then it's often the first one that is proposed to you by Zoom.
Oh, okay. Yeah, should be okay now, right?
Looks great.
Yes.
Oh, okay. Yeah, so basically my proposal is devoted to studying how glycation affects translation. And so there's some reasoning behind this, and I've looked at relevant publications. I came up with an idea. So what I want to start with is with this small note made by Francis Crick in 1956. So where he outlines what later became the central dogma of molecular biology. So information in a cell, it flows from DNA to RNA to protein. And for a cell to carry out its function properly, this flow of information has to be as precise as possible. However, it is not. And so whereas DNA replication is relatively precise, and we know that DNA polymerase misincorporates one nucleotide per 1 billion nucleotides, RNA polymerase during transcription misincorporates one nucleotide per 100,000 nucleotides of the template. So, and the process of conversion of information stored as a nucleotide sequence into amino acid sequence is even less precise. So a ribosome, it incorporates one amino acid per 10,000 codons. So this is actually a very crucial step, very error prone, that ensures the proper functioning of this whole information mechanism. And so when there's accumulation of those improper polypeptides, so we call it the loss of proteostasis, and it is a hallmark of aging. And we know that proper proteostasis is crucial for longevity, and several species that are known to be long-lived, like naked mole rats, they are known to possess a very accurate translation. Yeah, so we have to bear this in mind. And so when it comes to like energy producing processes inside the cell, so they start with a step called glycolysis. So it happens inside the cytoplasm. And so it's basically an oxidation of glucose to pyruvic acid. And during this process, there are intermediates called hydroxyacetone phosphate and glyceraldehyde diphosphate. And these compounds, they can spontaneously decompose into a compound called methyl glyoxal, which is highly reactive and highly toxic. And it is known to modify proteins and nucleic acids. So it can attack arginines, it can attack lysines and cysteines, and it's also been shown to attack guanine. And I have to stress that this process is completely stochastic. It is not mediated by any enzyme, and it is basically universal. And so everything started with this paper by Paul Tarnali and Naila Rabbani. So these are very prominent people in the field of glycation. So basically what they showed is that cells, when cultivated in high glucose, they tend to exhibit the so-called unfolded protein response. And so they conclude in their paper that proteins, when modified by methyl glyoxal, they lose their conformation and they elicit the unfolded protein response. So in my reasoning, I went a step further. So since we know that methyl glyoxal basically reacts with everything inside the cell, so it inevitably will react with ribosomes as well. And so we know that ribosome is a molecular machine which consists of proteins and ribonucleic acid. So here on the picture, you can see that, like, for instance, for 60S subunit, in light yellow, it is RNA. So there's a huge amount of RNA inside the ribosome, and dark yellow, it is protein. So there's a huge quantity of sites for methyl glyoxal modification. And so my hypothesis is as follows. So if a ribosome gets glycated, so what actually happens to it? So does it break down? So does it stop working? Or is its function somehow altered? And so what I have hypothesized is that methyl glyoxal, so it reacts with ribosome, either ribosomal proteins or RNA, and this is reflected in how the ribosome functions. So it is reflected in translation accuracy. And this modified ribosomes, they actually become less precise in translating mRNA and produce proteins that are prone to misfolding. And so I had an opportunity to do some preliminary experiments. And so I was able to experimentally elevate methyl glyoxal production inside cells. And so here are my results. So when I cultivate my cells with a chemical that elevates methyl glyoxal production, I can see that the amount of modification increases. And to measure the translation accuracy, I used a so-called dual luciferase reporter system. So in which one of the luciferases harbors a codon replacement inside its catalytic side. So whenever there's a misincorporation event, the functional enzyme is produced, and based on the luminescence readout, we can estimate the level of ribosomal misreading. And so I had very little sources, and I was able to measure only one concentration. And so my results, they were quite unexpected, because what I saw is that upon the inhibitor treatment, the translation accuracy is actually increased. So my results, they contradict my hypothesis. However, there could be many reasons for that, and I need to do additional experiments to see what is the reason behind the observed effect, and to be truly sure that the results that I obtained, they happen at the level of ribosome. I need to recapitulate my experiments and recapitulate my results using isolated ribosomes in an in vitro system. So so this is basically my hypothesis, and thank you all. And I want to express my huge gratitude to VitaDAO, who through their longevity fellowship funded my early studies. And and the results that I showed, that they were taking using their support. Yes, thank you very much.
Awesome. Thank you.
Thank you so much. All right. And now we have our third place winner. We have Shahaf Peleg and Andrew Wojtovic, and I hope I pronounced that correctly, from FBN, and they worked on external energy replacement. It was a really, really fascinating submission, actually. So we'd like to give them the mic and have them talk for a little bit about their work. Oh, you got Galadriel. Cool.
Yeah, so you get already extra point. So first of all, thank you everyone for coming here. Thanks. We would like to thank the judges, both the ones that picked our project and both the ones that did not, and congratulate the other two winners. I'm going to share with you some provocative hypotheses that we have been running through lately, and it centers around light as an energy replacement in our bodies. And I'm going to go through that. So first of all, maybe a starting point. This is a very nice provocative perspective that was published in Nature Aging recently, and you should really read it up. And two of the sentences that I chose to highlight are that taking all the science that has been done in longevity at the moment, it seems like that breaking through our biological limits, so around 120 years old, is impossible, which is kind of gloomy. And they said, however, if you really want to increase lifespan, then if you want to radically extend lifespan, then it requires a radical alteration of something very fundamental within our biology. And I think it echoes really nicely what I've been thinking recently. There's so much we can achieve with calorie restriction and drugs and so on. How about something more crazy idea to change our fundamental biology? That's exactly what we have been looking through recently. So if you look at this model human body, you can appreciate a lot of us is designed to seek food, eat food, and then manipulate it in some way so we can break this food, be it a cookie or ice cream and so on, and really extract the glucose. And we do it to transform energy. So we take the food to generate energy. And when you look now at all these macromolecules which are being reduced into glucose, this glucose finds its way to cells. Now, don't look at all the details. It just to illustrate that all this glucose transformation, what we call metabolism, is composed from many, many, many reactions, which work in some sort of a harmony. So Rakan actually touched this, the glycolysis. He actually said in some of the introduction. And the glucose endpoint is to generate ATP, which is the molecule of life. And I would like you to focus here on this part, which is in the mitochondria. It's called ETC, or the electron transport chain. And what this very, very conserved and fundamental mechanism of all eukaryotes basically is designed to transfer protons from one side of the membrane to the other one against the gradient. And then throughout all these complexes, enable enough protons to be on one side, only so they can go to the other side and make some sort of change in the rotation of this ATP synthase, which generate ATP. And this is very, very crucial a pathway, because if you block it, think about it, if you don't consume oxygen, you die. So you actually need to have it constantly on. So our bodies already know how to generate energy. Why even try interrupting with such machinery that have been evolved for 2 billion years of eukaryotes? So the problem is the following. There are a couple of problems with this, but one aspect that I came through with this project was the idea of the pollution. So when you take nutrients and you burn them with oxygen in the mitochondria to generate energy, you can use it as a metaphor to what our plants are doing. They take carbon, they burn it with oxygen, and they generate electricity, which is much needed in our modern society. But immediately what comes into mind when you look at these chimneys of these big plants, oh, they are polluting the air, right? So the idea is that the problem is when we generate energy, we also generate toxic molecular waste, which can be accumulated with aging and so on. And I'm going to touch it a bit later. So my idea was like, what can we do against it? And immediately what's coming to mind, well, how about we replace all these plants that burn coal, for instance, and we use solar energy? It's much cleaner, it's much better. So that's exactly what we wanted to do to us. And this is something that Andrew Wojtowicz, my partner in this application, and his student Brandon Berry looked in. So this is, again, this electron transport chain supporting protons to be used for ATP. And they did something incredible. They took a light-activated proton pump from fungi, and they engineered it so it will go inside the mitochondria. And what it does, it can translate the energy of lights using something called transretinal, and it can pump protons just via the light activation. And by doing so, it's partially replaced this very, very conserved electron transport chain. And this tool that was invented, and then we started to collaborate. And the hypothesis was that this will actually increase longevity. And I give you just now 3 points that we have in mind. First of all, if you use this MIT-ON, this light-activated proton pump, lifelong, you can probably generate enough ATP or enough energy, while doing so, produce less molecular waste, which can accumulate in aging, and it's irreversible. Another thing which you can associate with aging, you see an old person in the street, it looks like this person has less energy. If the mitochondria can be dysfunctional in this age. So this MIT-ON could potentially charge our energy with extra energy as we get older. And another thing we thought recently is, I guess, caloric restriction is very popular, but if you eat less foods, there is a maximum you can eat less because you still need to generate energy. And perhaps there will be some sort of synergistic effect by using our light-activated MIT-on. It will allow us maybe to push further caloric restriction while generating enough ATP. So this was the hypothesis. And as a proof of principle, I'm going just to show you a little bit of data. If someone wants to discuss it with me later, I'll be more than happy. So this is a paper that me and Andrew published recently. This is the proof of principle in worms. So if you take worms and you let them the ability to generate energy via light by activating MIT-on, you can appreciate that these worms have an increased mean lifespan and also maximal lifespan. So Andrew and I showed that this very unique idea can actually work, at least in worms. Now, when I go to conferences, a lot of people ask me, so, okay, worms, nobody wants worms or flies to live longer. What about mammalian cells, such as mice and human? And Andrew actually, in another lab of Minzu Kim, showed recently that it can actually also work in mammalian cells. So here you have HeLa cells, and you have also D8 T cells from mice. And you can appreciate, if you compare dark versus light, when you have MIT-on in mammalian cells, you can have an increase of ATP production. And this is great because it shows us proof of principle. We can use MIT-on mammalian cell. So in the future, in the outlook, what we would like to do is to really test if using this light-activated proton pump inside mitochondria can increase longevity in human, being a disruptive idea. And this is something we're actively pursuing. Maybe this is a good time to announce it, that we are in the steps to opening a company that will focus exactly on that, starting, for instance, from human skin. You can see the appeal that we can just use this gene therapy, have the lights that is outside or something which is a bit more house design, and we can generate energy in our skin. It's not going to completely replace, but it's going to be partially replaced. So it's going to be partially clean energy. And let's see how much we can push this technology forwards. And with that, I want to thank everyone in my lab and my amazing collaborating Andrew Wojtowicz, and really special thanks for the Impetus grant. They really fund crazy ideas and they allow us to do this. And I'm really, really thankful for that. Thank you very much for your attention.
Thank you so much. That was great. Yeah, we're a big fan of Impetus grants too. We love how quick they work. It's really amazing. So I just want to mention real quick before I hand it over to Stephanie Daynow, who's going to announce the honorable mentions, that the first prize was $20,000, the second prize is $5,000, and the third prize is $2,000. And it's just a small thank you from the community and from us. And we just want to support the work that you're doing. And again, we want to thank Vitalik Buterin and Gitcoin and all of the supporters of that Gitcoin round for helping to put together the funding for this initial kind of inaugural challenge of the Longevity Prize. So I'd like to kick it over to Stephanie Daynow now to tell us about our honorable mentions.
Hi, everybody. Thank you for having me, and congratulations to everybody. This is such a wonderful initiative, and I'm so thrilled to have Lifespan as part of the organizing crew in some capacity. We are here to try and make this movement mainstream. And with that, I won't delay. Really excited to provide some of the honorable mentions. We have Peter Hollins from Keygenics. And Peter, if you could give a couple sentences on your human, very small embryonic-like stem cells and longevity topic, that would be wonderful.
Yeah, sure. Thank you very much. And great presentations, guys. They were fantastic projects. Particularly interested in the embryonic one because I'm an IVF person. Anyway, our interest is in very small embryonic-like stem cells. They're found in platelet-rich plasma, PRP, and we work on the autologous PRP. And we use a modified laser, a red laser modified, which actually activates these stem cells. Now, in normal physiology, they're thought to be quiescent, so they're not doing anything much. And they are derived from the primordial germ cell. So what we have is the availability of a set of cells which are quiescent, and they've not really aged with the person because of their quiescence. And if we reactivate them with the laser, then you've got a source of somatic cells with a very young biological age. So this is our general concept, that we want to look at laser-activated VSCL stem cells in the anti-aging process. We use cgDNA methylation as one of our parameters, and telomere length is the other one. But there are lots of other things that we could look at. And our initial data shows that this does indeed reduce DNA methylation. The use of these cells does reduce DNA methylation. So that's important too. So very briefly, that's it. Thank you.
Thank you so very much. Up next, we have Mikhail Batten from Open Longevity. His title was Age-Related Changes in the Structures of the Extracellular Matrix. Well, if you could give a few words, and congratulations on being an honorable mention.
I think Mikhail couldn't make it today.
Couldn't make it? Okay,
to
we'll
Christian.
move forward. Is Christian Schaefmeister here?
Yes, I am.
Hello. Well, congratulations. You're the next honorable mention coming from Temple University. Your topic was rewinding ages with ligozymes, if I pronounced that
Yes.
correctly. I would love for you to give some information about that.
Sure. So very briefly, as we age, the idea is that the extracellular matrix gets rearranged and cross-linked as glucose reacts with the proteins in the extracellular matrix. And hold on, I just realized I'm not sharing my screen here. So sorry about that. There we go. Okay, everybody see that?
Yes.
Everybody see that? Yeah. Okay. Right. So very briefly. So sugar, glucose reacts with extracellular proteins, creates these crosslinks that we as we age, they grow in our extracellular matrix, make all of our tissues more stiff, and we have no natural mechanisms for breaking these down. So so sugar, proteins, time creates these crosslinks, and it's this fused ring system that forms between lysines and arginines in the extracellular matrix. There are people looking for enzymes, bacterial enzymes that could cleave these things. But the problem is there's a couple of problems. One is the enzymes are large, and to be able to fit into the very tight spaces where these crosslinks are, they're going to have a lot of trouble getting in there. Also, we don't have any natural proteins that do this. Bacterial enzymes, if you inject them into people, will cause a violent immune response and cause a lot of problems. So what I was proposing is that we create catalysts based on the molecules that we are developing in my group and my company to cleave these crosslinks, to recognize them and cleave them. And that catalyst would have to be small, about 5,000 Daltons. And we would discover these catalysts by making a whole bunch of them and then treating them with a special substrate that we would create by synthesizing the glucosapane crosslink that's separating a fluorescent quencher and a fluorescent group. And then if you discover a catalyst that cleaves it, then that would light up and you could detect the active catalysts. So the proposal is to make what are called DNA encoded libraries of catalysts. So linking 4 of our spiroligomer segments together to make a molecule that's big enough to wrap around a glucosapane with a DNA code attached to each one of them so that we could identify which ones are the active catalysts, to make something like 7 billion of these catalysts and then screen them for the active catalysts. If you have 7 billion sort of molecular boxes, you will find things. The molecules are based on our chemistry. They are these very large, complex molecules that we have made 2.5 million of these molecules in the last 3 years in my company. And we are developing these the molecules currently to bind proteins, but to develop them to make catalysts is something that I was proposing here. So I will end there, both my group and my company, developing this technology. And I'll stop there.
Thank you, Christian. Yeah, fantastic. And one last honorable mention is James Woodsworth. He's not here today, but he's from Newcastle University, and his title is Selective Destruction Theory of Aging. I just want to say
I'm actually here.
Oh, okay. Please take a minute. I know we're running late, so go for it. And then after that, I'll close out and hand back to Allison.
Yep. Hi, I'm James, not Bob. I'm on my father's computer. Couldn't change it. So if you imagine, for example, a cell that has undergone a mutation to gain faster metabolism, so it grows and replicates faster than the surrounding cells. Well, I mean, what are the consequences of this cell? Firstly, you'd expect it to compete the other cells in the tissue and become perhaps even the dominant cell type within that tissue. It would have a faster mutation rate, so you expect it to be more tumorigenic. And it could be associated with a host of other organ or specific or more general overactivity disorders. For example, beta cells that are more proliferative produce more insulin, and you could die of hypoglycemia. Fibroblasts will induce fibrosis more easily if they're more proliferative. So basically, these cells are very dangerous. And what I was essentially suggesting is that organisms could then gain a survival advantage if they, instead of pushing cells towards these very dangerous fast cells, had a mechanism which would allow the slower cells to basically slow them down epigenetically. And essentially, the result of that would be metabolic slowdown, which is essentially what we see with aging. We get insulin resistance, matrix degradation, cells replicate slower, as Shahaf has already said. Basically, we have less energy, and that's probably what aging is.
Got it. Thank you so much. And again, I just want to say congrats to everybody. We're really happy that this prize works against siloing information and the calcification of ideas by allowing everyone to come together and build in public. And this is becoming the spirit of our field. So congrats to everybody. And thanks everyone for having all of your organizations be part of this and ours as well to represent. And Allison, please take us to the end.
Yeah, I just want to congratulate all of you guys. And again, thank you so much for everyone who judged the prize proposals. And of that, Shahaf pointed out all the judges that judged for and against them. We seriously appreciate all of your efforts. Thanks to everyone who's on the call from the judges. It was really, it was much appreciated, all of the work that everyone put into this. I do want to kind of like just close by saying that this is one of many prizes. So this was the first one, the Hypothesis Prize. We have many other prizes in the making. A few of them were already pointed out by Dane at the beginning. There'll be plenty more to come. And this was like a really great first kind of like trial run of this entire endeavor. And it worked fantastically. And so really excited for future prize suggestions, also donations to the prize if you're interested in really pushing this field forward. Feel free to suggest or even support financially one of the prizes that come out of this. That's all possible through the website, longevityprize.com. And we really look forward to like driving this entire effort in a collaborative spirit. So thanks everyone. Thank you again to Vitalik and all the other donors who made the first one possible. Thanks to everyone who participated in the first round. And we can't wait for future prize proposals. I look forward to seeing some of you in person at the longevity workshop that we have, including our winner, Carlos, where you have a little bit more time to present. Thanks everyone for joining this virtual announcement. And that's all for me. And unless Dane or Vincent has anything else to offer, I just want to leave it with a big congratulations to everyone who's made this possible.
That's great.
Yes, not much to add other than checking out our document to propose your prizes or comment on another ones so we can add some new prizes.
There we go.
Okay, thanks. Thanks, everyone. I hope you have a wonderful rest of your day. You'll be hearing much more in the proper press announcement and so forth. But this was very joyous. I'm really excited. And I'm hoping that this will really advance the field a little bit more forward because we all depend on it. All right, everyone. Have a wonderful rest of your day. Bye-bye.